Although genome editing can sound quite complicated, the basic principles are relatively simple. The main use of genome editing is to introduce mutations in very specific points in a chromosome. The most common technology used for genome editing, and the youngest one, is called CRISPR. CRISPR uses two different components. One of them is a molecular scissors, and the second one a homing device. The molecular scissors is a molecule of a protein that we know as Cas9, and the homing device is a molecule of RNA, also known as guide RNA, because it will guide the Cas9 scissors to the right place. So let's assume that you want to introduce a mutation in this specific point in a chromosome. All you need to do is to design the homing device in such a way that it guides the whole complex to the right place in the chromosome and cuts the DNA. As a result, you will have a break in the DNA, and that is not an uncommon thing. Actually, it's a lot more common than everybody believes. It happens in humans, it happens in plants, it also happens in animals. It's so common that we do have a repair mechanism in our cells that will come into position and solve the problem, leaving the DNA as new. But these repair mechanisms sometimes make mistakes. It comes into position and the repair is not perfect. When that repair is not perfect, it introduces a mutation, and that is exactly what we want to do with genome editing. Now, genome editing is only the first part of this equation. It's the ability to make cuts into the DNA at very specific points. The rest is performed by the machinery that everybody have in their cells.